To finish out Chapter 5, we're going to talk a little bit about epidemiology and epidemiological studies. If you are not familiar with epidemiology, it literally translates as the study of what falls upon a population. But this is a field of science closely related to medicine and hence to biology, where epidemiologists are basically trying to understand how particular diseases are transmitted, make links between maybe specific environmental conditions and diseases, things like cancer, and in terms of infectious diseases, trying to help establish effective control measures that are going to interrupt the transmission of a particular virus. So, critically important in times of a pandemic like COVID-19 now, you're constantly hearing interviews with epidemiologists or at least seeing in the news what epidemiologists are reporting. So some terminology that epidemiologists would use and would pop up in epidemiological studies. We can talk about disease occurrence being sporadic, endemic, epidemic, or pandemic. So sporadic, of course, means infrequently, so there's no particular pattern to the disease. It pops up here and there without any consistent pattern. Endemic disease is referring to sort of the normal background amount of a particular disease in a population at any given time, right? So, throughout the year in the United States, there's going to be X number of people that have the flu, right? Even in the middle of July, there's going to be some people that have seasonal flu. But of course, we get bigger spikes of flu in the winter. We'd say it's a flu epidemic, right? Epidemic being a localized outbreak. And then if that outbreak is global, then it becomes a pandemic. And a big focus of epidemiology is what's called the triad model, which is this idea that you need these three things working in concert together in order to cause disease. So of course you need an infectious agent, virus in this case. You need a susceptible host. Right? I've already talked about that interaction a little bit. But then the third corner of the triangle is the environment. So you need an environment that is going to bring the infectious agent and the host together in a way that allows disease to happen. And we can kind of zoom in on that and think about this in terms of some of the other topics that we've talked about in terms of what we're going to call here the chain of infection. And so if we think about this, the infectious agent is going to be transmitted to the host when the agent leaves what we call the reservoir, which could be another infected person, could be an animal, could be the environment, but leaves that reservoir through a portal of exit. We already focused on human portals of exit. Is going to get conveyed by a mode of transmission to another susceptible host over here. And of course, we've got different modes of transmission. Could be vectored, could be through droplets or airborne and aerosolized particles, through direct contact, through some other vehicle. And we're talking about human viruses here. Humans are going to be the most common reservoir of human viruses. And there are some viruses that have been identified that humans appear to be the only reservoir. So poliovirus, variola virus, basically the cause of smallpox, apparently only infect humans. Whereas a lot of other viruses may be considered zoonoses, meaning they can infect other animals and the virus could be transmitted from those other animals to humans. So influenza, this is known to be the case. Famously with the 1918 influenza epidemic that killed, millions of people worldwide, that was believed to be transmitted from pigs to humans. Ebola, and now with MERS and SARS and SARS-CoV-2, COVID-19, appears to be able to be transmitted from bats to humans. So you're probably familiar with this term being a carrier. Carrier is really any reservoir that can transmit the pathogen but shows no symptoms. So that could be a human, an asymptomatic human, or an animal, right? A lot of these viruses may not make animals sick to the extent that they do humans. So they'd be considered carriers. And I already sort of briefly went through some of these modes of transmission, but in general, we could consider transmission to be either direct or indirect. So direct is going to be through direct physical contact. We would actually consider respiratory droplets to be a form of direct transmission because they stay airborne for such a short amount of time and are usually going to be found close to the individual that produced those droplets, whereas indirect transmission requires some intermediary. And interestingly, aerosolized particles we consider indirect transmission because they stay airborne for such long periods of time. But could it be other vehicles, which would be non-living physical substances, food, water, blood, fomites, which would be something like utensils or something like that that might have virus on them, or furniture. Um, and then vectors are going to be distinguished from vehicles in that they're the living intermediaries. So insects, for example, uh, have arboviruses, which are arthropod-borne viruses transmitted by mosquitoes, fleas, ticks, etc. Another important aspect of epidemiology would be, as I mentioned earlier, how to break the chain of infection, right? And we do that with control measures. So, we're all too familiar with some of these from learning about COVID-19, but quarantine, right? Patient placement, isolation, PPE, we all know what that term means these days, masks and gloves, etc. Personal hygiene, right? Hand washing, very important uh, for many viruses to break that chain. But it could also be something more complicated like environmental infection control, right? If the infection is being easily transmitted uh, through the environment, how can the environment be cleaned or sterilized, or how can we prevent people from going into that environment to pick up that virus? And then, of course, monitoring and management is an important part of this as well. So, like contact tracing, for example, would fall into that category. And the example I give you here is basically quarantining people that had been in contact with MERS, a virus closely related to COVID-19, quarantining them for 14 days and trying to figure out who they've been in touch with, etc. But head and shoulders above the rest, by far the most effective control measure is going to be vaccination for in terms of virus control. And we're all familiar with vaccination, right? You're basically introducing either non-infectious parts of a pathogen or potentially a whole pathogen that's been modified so that it is no longer infectious into an individual so that you mount an immune response against that virus. And then if you do encounter the actual pathogen, your immune system knows how to handle it very quickly. And the big advantage of vaccination is we can get herd immunity in a population if vaccination rates are high enough. And so I know we've all probably heard this term, but again, for herd immunity to work, you need a high percentage of the population to be vaccinated. So on the left, we're seeing an example where herd immunity is working. On the right, where people have been vaccinated, but we don't have enough vaccination to get that herd immunity. And the idea here is we want to protect unvaccinated individuals, maybe people who, can't be vaccinated because of cost or don't have access to health care, or maybe they have some other health reason for not being able to be vaccinated. But the idea here is, infected individual enters the population, everybody in yellow has been vaccinated. We do see limited localized spread, right? But the spread is not able to reach the majority of the unvaccinated people because they are surrounded by, large numbers of people who are vaccinated and are not going to be spreading the virus. Whereas in the situation on the right, we just don't have enough people vaccinated to offer that protection. So yeah, the individuals who are vaccinated are safe, but unfortunately those that either didn't get vaccinated intentionally or weren't able to be vaccinated are still at risk. So that's going to be the big challenge going forward with COVID-19, assuming we do come up with an effective vaccine, is trying to get it distributed and get a large enough percentage of the population to take the vaccine to offer that herd immunity. Now, I wanted to finish out here just talking briefly about epidemiological studies. So we talked about epidemiology as a field, the kind of things that would fall under that umbrella. But how do epidemiologists actually conduct epidemiology, conduct their science? Um, and so there's a few terms to get out of the way here. Epidemiological studies would be concerned with things like morbidity and mortality, right? Morbidity being the rate of illness, mortality, obviously, the rate of death. They would talk about incidence versus prevalence. Incidence would be the number of new cases that occur in a population during a particular period of time, whereas prevalence would be the total number of individuals that are infected at a very specific time. So we could say something like this. In 2012, the incidence of HIV in the US was reported to be 18 cases per 100,000 people, while the prevalence was 342 cases per 100,000 people, right? So the 18 is a smaller number because that's just dealing with new cases, whereas 342 would include people that were infected 10 years ago, right? But that are still alive. And another important point to make when we think about studies is before we could actually start counting cases and figuring out who's sick, where are they sick, is we actually need to know what qualifies as being sick, right? What is the case definition is the technical term. So these case definitions are a well-defined set of clinical and lab criteria that are going to be used to determine if a person has a particular disease. So I'll give you one example here, which is the 2013 case definition for measles. And you can see there's a clinical description, right, which talks about specific symptoms, right? You have to have this macropapular rash lasting a certain number of days, temperature over a certain amount, cough, etc. And then we have case classification is always going to be either probable or confirmed. And probable is saying, okay, well, we have some of these symptoms, or all the symptoms maybe in the clinical description, but we don't have any definite linkage or laboratory confirmation, right? So we can't say, oh yeah, this person's husband or wife was diagnosed with the disease. They haven't been lab tested, but we think, well, maybe they have the disease. They've got the rash. Looks like it's probably measles. Whereas confirmed, you've probably got the epidemiological linkage. Again, we know this person came into contact with somebody with the disease, and then we've got some lab testing, either antibody testing or PCR testing, whatever the case might be, showing, yep, they do have that particular disease. And there's some tentative case definitions available for COVID-19, but because it's so new, I wanted to focus for the purpose of our lecture on one that was more well defined. But you could look that up online if you're interested. Now, in epidemiological studies, there's really going to be these 5 W's that epidemiologists will consider. So it's who, what, when, where, and why, or how. So some of these are obvious, like who is, what kind of people are getting infected, how old are they, what's their sex, socioeconomic status, all that would fall into the who. What would be the case definition we just talked about, right? What are the symptoms? What kind of testing do we need to do to confirm it or consider it probable? Where, that's self-explanatory. when would be, time of day, time of year, etc. And then the why is really more complicated, talking about, okay, well, what are some of the risk factors? What are some of these modes of transmission? And really, this is where we get into some like more analytical type studies that would be needed to determine the why. Okay, so these are all considered epidemiological variables. So, just to give you an example of when we think of influenza, right, flu season, we're all aware that that peaks in the winter months, right, basically starting maybe in November, but really getting underway December, January, February, and then by March, things are usually starting to tail off. So there are some exceptions to that, but that's pretty typical of a flu season, at least in the northern hemisphere. It's of course just the opposite in the southern hemisphere, where their winter is in our summer months. But if you're an epidemiologist looking at that, then you start asking, okay, well, we get this defined pattern. Why is that the case? And I don't think it's been totally determined, but there's a few educated guesses. Data suggests that drier weather in those winter months may help disseminate aerosolized particles. They stay in the air longer, can travel farther. So that's creating a higher risk environment. And then you've also got people spending more time inside around other people, which is going to increase transmission as well. And so we likely have similar concerns with COVID. I think there was some hope that with COVID it was going to decline in the summer like we see with influenza, but unfortunately that doesn't seem to be the case. So it may just be burning hot and heavy and steady through the entire year, or it's a bit worrisome to think we may see an even increased peak in the winter months again as well. So then with the why, again, this is really where you need some type of analytical study. And so I just wanted to talk about the two basic types of epidemiological study designs. So they could be experimental or they could be observational. And experimental means it's going to be planned. We would consider that an analytic study. So an example of an experimental epidemiological study would be like a clinical trial testing a new vaccine, where you're putting test subjects into specific groups. There's a study coordinator that is assigning people to these different groups. Whereas observational is not really analytical because you're just observing subjects and recording the data, right? So if we go back to the vaccine example, okay, let's just look and see, who got the vaccine versus who didn't, and what are the outcomes in those two groups. But we're not influencing who gets the vaccine or who doesn't. So, in observational studies, again, there's some subtypes: cohort, case control, cross-sectional studies. And I just said these are not usually going to be analytical, but actually 2 out of these 3 types can be analytical. Cross-sectional studies, which are really the most descriptive, tend not to be analytical. But just to show you a brief description of differences between these 3 types here. So cohort and case control studies are both controlled, meaning you have a control group, which makes them analytical. In a cohort study, you've got 2 groups that are being compared in real time, whereas case control studies are always going to be retrospective, meaning you're looking at events that happened in the past, right? Looking at data that's basically already been collected from individuals, and you're assembling that control group from people that happen to be in the same place at the same time, that didn't contract the virus. Whereas, in the cohort study, because it's in real time, you have a control group from the get-go moving forward, but people are not being specifically assigned to those particular groups, right? We're pulling those out of the existing population. And as I mentioned, cross-sectional studies tend to be almost entirely descriptive. So, as that name implies, we're just taking a random cross-section of the population and analyzing the data, looking for correlations there. And this is where epidemiology can sometimes get itself into trouble because it's pretty good at finding correlations, but of course correlations don't always mean cause, right? Correlation does not equal causation. So there's all kinds of examples in the literature of epidemiological studies suggesting that this is maybe linked to that particular disease, or something in the environment might be linked to cancer, but it's actually pretty tough to establish causation with these cross-sectional studies. So if epidemiology is something that you're interested in, we actually have an entire course in the biology department on epidemiology that you might look into taking in the future.